Thank you for inviting me. Okay, slides. So I'm going to tell you a little bit about what we're trying to do now at National University of Singapore. I've been there for about 6 to 9 months, and I'm going to mix that in a little bit with some talk from data from my own lab, mostly from my lab in California, which I still have at the Buck Institute. I think that, well, I can tell you a little bit of the reasons that I went to Singapore later in the talk. I'm hoping it can be a real a site for testing interventions in human aging. That's really the goal. This has sort of been my career trajectory and during research, and it's kind of been where the field's been going too. If you look back at aging meetings like 3 decades ago, most of the talks, or many of the talks, were in these non-vertebrate models, yeast, worms, flies. That's where I got my start in yeast and a little bit in worms. They turned out to be great model organisms for trying to identify the range of genes and pathways in an unbiased way that could affect the aging process. And that led us with lots of different candidates that might affect aging. A lot of the last decade has been spent trying to test which of these pathways are functional in regulating aging in mice, and surprisingly a lot of them are. Other people are looking at primates and dogs, and I think now we're at this point where we're really ready to start trying to validate things in humans. And the question is, how do we take interventions, no matter what kind of intervention they are, and test them in some effective way to see if they're altering human aging? I don't need to tell anybody here that the goal is health span and not just lifespan. But how do we measure this? What does health span mean? And in a world where we don't want to do trials for 40 years, how do we measure lifespan? And I think that one strategy that's emerged really is to try to develop targets or biomarkers as diagnostics to measure whether these interventions are working. And I'll talk a little bit about that. The other is to really try to prevent early stage disease progression and things like the Tame trial or variants on that. I'm not sure which one is right, but I think we have to start testing in this area to begin to understand how to design these studies, and then eventually the idea is to implement this in a way that we're actually doing healthcare instead of sick care, which is what most of the industry is doing right now. Now, if you look at the lifespan extension agents out there, there really is a growing list. This is actually woefully outdated at this point. There are many different behavioral interventions that are thought to affect aging. And also many different small molecules. And a lot of what my lab has been doing right now is to try to take a harder look at these different interventions and try to understand mechanistically how they're impacting the aging process. We've worked a lot on rapamycin, and I'll just mention that briefly today. But the lab's also worked on metformin and ibuprofen and acarbose and a range of other things. We have a couple other things to put on this list that aren't published yet that I'm not going to talk about today. And I think that the understanding them mechanistically is really important because it'll give us a better feel for how they might work in humans. And it'll also tell us which ones can be combined together to have synergistic effects and which ones might cancel each other out. And we have some evidence for both now when we're trying different interventions simultaneously. Of course, the behavioral interventions have been tested in much more detail. Calorie restriction, there's great data on intermittent fasting, and I thought I would start my talk today in honor of Aubrey by discussing alcohol a little bit more in depth. So there have been many studies on acute toxicity of alcohol in animal models, but much less looking at whether you can draw out the benefits of alcohol with low to moderate consumption. And the reason I got interested in this is the National Institute of Alcohol Abuse and Alcoholism invited me to give a talk on the benefits of alcohol and aging. And I was a little bit worried about people throwing fruit and vegetables at me, but they were actually nice. But I went through the literature, and there's really surprisingly little done in animal models, and we really don't know what the mechanism of the benefit might be. And so let me dispel a couple myths. Well, first let me show you the recent metadata from the human study. So this is about 33 different studies put together, and we wrote a review on this. This is the actual, where we took this graph from, and we also wrote a recent review. You really have to consume a lot of alcohol before mortality rate goes up beyond 0. So this is abstainers here, and for men, you have to get almost to 4 drinks a day before you're got back to where you were at abstainers. Anywhere in this gap means you have a lower mortality rate. Women, the same is true up to 2 drinks a day. So there really is a striking tolerance for alcohol consumption. And it's not really red wine, much to the chagrin of people in Napa Valley. If you do look at cumulative hazard here, for people that have high wine consumption, they have a lower cumulative hazard, so it appears that there's some benefit from wine. But if you factor out socioeconomic status, there's really no benefit. It turns out that rich people have lower mortality rate, and they also like to drink wine. Now, there have been a lot of studies looking at potential benefits of moderate alcohol consumption. And there are also problems with even moderate alcohol consumption increases risk as well. So there are benefits in cardiovascular disease. Surprisingly, there's data out there suggesting lower risk of diabetes, metabolic syndrome, potential benefits in cognitive disorders, even protection against weight gain, which is surprising given that there are calories in alcohol. But there's also a higher cancer risk for digestive cancers, some lung type tumors, and breast cancers. So not everything is good here. I think like all aging interventions, you're going to have many benefits, but also some increased risk. And as I said, I'm going to go through this fast. Ethanol is a source of calories. And so it's really kind of striking that it might provide protection against overnutrition or diabetes, because you're adding extra calories, really kind of empty calories into the system when you're drinking. So we've done just one pilot experiment, but I just wanted to show you the results just to give you a little bit of a feel for where we're going on this. And so we took 10 mice and put them on 10% alcohol in the drinking water, starting at 5 months of age. I'll get back to that, just relax. Starting at 5 months of age, and then at 10 months of age we did some metabolic analysis and then harvested the mice at 15 months of age. Now 10% alcohol in the drinking water sounds like a lot, I agree. But I want to tell you that it's not that much when you think about it. This is the California advisory panel for how much blood alcohol content you have per alcohol consumption. They recommend drinking 0 drinks a day, not surprisingly. But remember, when people are drinking alcohol, they're consuming a couple drinks a day in a short period of time. Their alcohol rate goes up and then goes back down. Now, the mice, we're not going to give them 2 drinks a day. It's too difficult, so we're putting in the drinking water. But mice also have very fast metabolism of ethanol. And so if you average this out, they get about the same overall level of alcohol that humans do, except it's dispersed throughout the day as opposed to going up and going down. So these mice still consume the water that have the alcohol in it. They still eat the same amount of food. They're actually eating, if anything, maybe perhaps a little bit more food. Their changes in respiratory quotient are almost exactly what we would predict if they're consuming alcohol, because alcohol itself changes the respiratory quotient. I'm not going to go into that in too much detail. And something that's true in humans is also true in mice. If they drink all night, they sleep during the day. And so the amount of sleep goes up, their exercise goes down. Their body composition is really not altered at all over time, either with raw or as a percentage of body mass. But surprisingly, they're protected from weight gain. And so we're still trying to understand what this means. We're trying to figure out where the calories are going. But this recapitulates some of the human studies that you can actually protect animals from weight gain by giving them alcohol. And I can't believe I'm going to say this, but their livers are protected too. Again, this is low level of alcohol consumption. High level of alcohol consumption is really bad for the liver. Don't get me in trouble. But in a 10-month study, there's much less lipid accumulation in the liver with a low level of alcohol consumption. So what we're trying to do now is get back into the real question, and I don't have any data for this yet, but what is the mechanism? Why is this beneficial? And one of the studies that's out there is that alcohol can actually impair the TOR pathway. So you can decrease muscle protein synthesis, which would be indicative of reduced TOR signaling, and you can directly affect phosphorylation of mTOR kinase targets with alcohol. And this is one of the things that so far our evidence suggests this is true, and this is one of the mechanisms we're looking at for a possible benefit. So more on this later, but so far, at least in a one-year study, we are able to recapitulate some of the benefits of alcohol in animal models. So hopefully this will lead to more mechanistic analysis. And I just want to touch on the TOR, and then I'll get to Singapore. So this mTOR pathway, probably all of you have heard about this now. This is one of the major flagship pathways modulating aging. The drug rapamycin extends lifespan and everything from non-vertebrate models to different kinds of mouse strains. It can be given to mice at early in life, late in life, for a short period of time in life, and it extends lifespan. So there's a very robust effect on aging by inhibiting mTOR signaling. And we've been working on this for a long time now. And our model is this, that whatever's happening with normal aging is not necessarily TOR. TOR is the response to that. So you get your favorite type of damage, perhaps that happens during aging: DNA damage, protein misfolding, telomere shortening, oxidative damage. And the cells respond to these accumulation of macromolecular damage by trying to upregulate the TOR pathway. And in this case, though, it's not a compensatory change, it's a maladaptive change. And this is further contributing to the pathology that's associated with aging. Now, it's striking how similar this is to disease. If you have a favorite disease that you study, go search on PubMed and look at TOR signaling. I'd say there's about a 50% chance you'll find a paper saying there's apparently upregulated TOR signaling in your favorite disease. So I think if we're looking at this paradigm here and trying to understand what kind of pathology is emerging, I think the disease state and the aging state are both going to contribute to elevated TOR, and all three of them in concert are going to define the type of pathology that's occurring in a given tissue or organ. Okay, so this hypothesis is somewhat bearing out in stem cells. There are now 4 different types of stem cells that have been shown to be, number 1, depleted with aging in terms of the ability to regenerate tissue, and 2, have elevated TOR signaling with age. We've just now shown this recently with a collaboration with Henry Jasper and Tom Rando in tracheal stem cells and also in muscle stem cells. It's been shown previously in hematopoietic stem cells. And there's another paper in mesenchymal stem cells. So we're really considering the model now that one of the ways that the TOR pathway is driving aging is through impairing regenerative potential in various adult stem cell populations throughout the body. And just to give you an idea how we're testing this, we're using a lot of genetic mutations in the TOR pathway, and now we can turn up TOR signaling, turn down TOR signaling, and to a simplistic level, what we find is you can really kind of ramp up aging or delay aging depending on which way you go in this pathway. And I just want to point out that I'll show you just one slide where we're looking at a mice that lack TSC2, and this is an inhibitor of TOR, so this turns up constitutive TOR signaling in the animal. Now, in a young animal, this is beneficial, and so what we see is that this is a paper by Marcus Rueg and Mike Hall, that if you look at a denervation reinnervation model in muscle, that if you have elevated TOR signaling, the muscle repairs faster. And if you have reduced TOR signaling, it takes longer for the muscle to repair. So you need elevated TOR activity to drive stem cell differentiation to repair the damaged tissue. And that's also true if you take out myoblasts and plate them. This is data from Tom Rando showing that these cells that have higher TOR activity start proliferating much faster in a dish when you pull them out. So these stem cells are poised to replace damaged tissue. This is the trachea now, and what you see in a normal mouse is that this is the epithelial lining of the trachea. These are the stem cells here in green, and they decline quite rapidly with aging. And what we can show is that if there's elevated TOR, if we impair TOR signaling, we can rescue that phenomenon. So this is, you can start rapamycin at different stages, you get similar results. Let's just look over here. This is the loss of stem cells, and we can regenerate, or we can recover most of the stem cells with rapamycin treatment. So this is really where we're looking a lot now with respect to TOR signaling, and this is the model that we have, that you need TOR activation for a quiescent stem cell to become activated and differentiate to replenish many tissues. Over time, though, with things that may be happening to these stem cells, you're getting an activation of the TOR pathway. It's getting too high in the stem cell population, and then you're getting too many cells that are going into a differentiated state, and that you're depleting the stem cell population over time because not enough cells are going back into a quiescent state in the aging animal. you can suppress this with rapamycin. So that just gives you a flavor of where we're trying to go. Let me tell you a little bit about Singapore. This is Singapore. Come visit. It's a little bit humid, but otherwise a nice place to go. And what we're really trying to do there is to establish a clinical approach to human aging. We're still very excited about bringing in preclinical scientists as well, but it's how do we take all this knowledge we've accumulated about aging and interventions and begin to apply it. And it's an interesting challenge when you think about it, because there's really two different ways that people that think about treating aging work. So you have the aging researchers who would be, enriched in this crowd, who are really thinking about preventing aging. What can we do to people when they're at risk of chronic disease? So again, if you imagine this is when you're born, you're healthy, then you start getting sick, and eventually it gets to be too much and you die. The aging researchers are largely looking at interventions here to drag out this period of health span, perhaps compress morbidity, so lifespan will be extended, but health span as well, and this is mostly done in animal models. If you look at what the geriatricians are doing, they're more about managing aging. They're taking people that come into the hospital in their 70s and 80s, they have multiple diseases, they have polypharmacy issues, and it's not a question of stopping aging, it's about how do we manage it in a way to improve quality of life as long as possible. And so the question is, if you're going to go into the clinic and try to take this approach, who do you work with? And so one of the challenges we have is to try to bring clinicians together with the researchers so that we're all thinking about things in the same way and taking a preventative approach. Obviously, both of these are important, and ultimately the effective strategy is going to be delay aging as long as possible and then manage it when you have to. But right now we have two different groups of people, the PhDs and the MDs largely, that are talking in different paths, and we have to bring them together. And so the goal over the long term is to really extend human health span in Singapore, identify and de-risk interventions, test them at healthy aging people or at least very early stage disease people, and then develop implementation plans. And one of the benefits about Singapore is it's a very progressive minded government. It can think 10 years ahead. It's probably still going to be there. And they're very concerned about aging. They have about the third longest lifespan in the world by one measure. The birth rate is 1.3 per couple. There's not much room to put more immigrants. And right now there's going to be 2.1 working people for every retired person 12 years from now. So the government is saying, what can we do about this? And they get the idea that it might be beneficial to really extend health span. So this island could essentially be a testing ground for interventions to really try to keep people healthy longer. So we're looking at different kinds of strategies right now. We're just getting up and running. This is the kinds of things that Nir Barzilai has been doing. This is the Tame trial, where you're looking at a long intervention trying to prevent many different diseases simultaneously. I think this is an interesting trial, and it may ultimately be the right one, but right now what I want to do is compare multiple interventions to each other, and I can't spend $60 million per intervention to figure out which ones are going to work in this context. So I don't think we're going to look at this. We're more interested in these kinds of things. This is what Nir's doing, although we may not doing quite this way, where we do short-term treatments and really begin to try to look at biomarkers of aging. I think that everybody knows that interventions have really become, a lot of them have been discovered in the last 10 years that might affect human aging. But the other thing that you'll hear about later this afternoon is that the biomarker field is really being revolutionized with regard to aging. So there's a lot of different potential biomarkers now. We don't really know which interventions are going to affect which biomarkers. We don't really know if they can just be slowed down or reversed. There's a lot of questions that remain open, but I think we have to begin to test these things in humans if we're going to know the answers. There's a lot of other emerging approaches beyond the ones I mentioned. There's nutritional supplements, young serum factors, senolytics. I'm not sure I'm ready for this yet, but there's gene therapy. Even mindfulness, there are a lot of studies in Singapore looking at community-based mindfulness, looking at yoga, looking at choral singing. I actually told the person if I'm having to do choral singing every week, I'm not sure it's going to make me live longer. It might drive me crazy. But anyway, I think the issue is there are lots of people out there thinking of different strategies to affect aging, but we have no way to really compare them side by side and figure out which ones are likely to be the most effective. And I'm kind of agnostic to the approach right now. I think the idea is to develop a strategy for this kind of comparison. And there are a lot of potential biomarkers about which you'll hear some later today. epigenetic algorithms, immune cell populations, metabolomics, microbiome, senescence. I can go down the list. I think that probably no one of them is going to be perfect, and what we're looking to do is create a suite or a platform of different interventions that we can apply to a blood sample and a stool sample, and then work across all kinds of different clinical trials to try to compare notes on how things are coming out. So this is generally the idea. You've got all kinds of different things you can test. You've even got like cardiovascular disease trials. The primary endpoint doesn't have to be aging. We can layer this platform of integrated biomarkers that we're trying to develop on any of these different interventions, and then really begin to hopefully get some data that's relevant to human aging in a large-scale way. Okay, we're also, I just mentioned this, establishing long-term cohorts in Singapore. One of the other advantages of Singapore is that it has 3 different ethnicities that represent 65% of the world population that are basically largely not tested. So we do know something about aging in the Han Chinese population, but if you look in the Indian and Malay populations, we really don't understand aging at all in those groups. And if you look at something like diabetes, there are very different kinds of diabetes that are going up in these ethnicities. There's a non-obesity related diabetes in the Chinese and Indian population that's really accelerating, and nobody understands it. I would be shocked if we don't see big differences with aging as well. And so I'm going to just stop there. I think that I'm very happy to talk to anyone who's interested in more in what we're doing in Singapore. We're recruiting faculty and postdocs and all kinds of things, and I think hopefully it'll be an exciting place to test some of these interventions that may affect human aging. Thanks for your time.